Yes. Go on.
Okay, you are welcome to the new normal. We are now living in the new normal, and so we have to adapt to what is going on around us. And what obtains now is that virtual lecture is also coming into play. So we have blended lecture. Sometimes you're going to be invited, and sometimes you're going to listen to some of your lectures online. So my name is Akintunde Olushola, a technologist in biochemistry department, and this is BCH 212. Today we're considering protein and amino acid, and then we just want to let you into how you can identify qualitatively some amino acid and some proteins in samples. This is very, very important in biochemistry. Proteins are large biomolecules that have chains of amino acid residues, and they are very, very important in catalyzing reactions and replication of DNA, and in so many other ways, functions that they do all over the body, just like some other biomolecules. Very, very important. So we know that this is one important topic that you have to be conversant with. So we're going to look at some qualitative tests that we can use to identify amino acids and proteins in samples. I'm going to name some of them. Just listen, and then you will get them. We're also going to practicalize how we can identify some of these in samples. So just listen and look and make sure that you take your notes. Let me also tell you, remember how we do our reportings in our scientific notebooks. Please make sure you write the aim, the apparatus, the methods, and then you write your results. We will tell you later how you are going to write these results. It's a very simple one. Just look at it, and then I believe that you're going to get it. So going to our fundamental test for proteins and amino acids. Number one, our fundamental test we're going to consider is our Neidrin test. And this usually is positive for most amino acids. It is positive for primary amines, primary amino acids, and then even ammonia, which is not a protein, which is not an amino acid, but is similar to amines because it has hydrogen is attached to nitrogen. So what is the color we expect when we are testing for Neidrin? We are using Neidrin tests on some protein samples or on some amino acids. We expect a blue or purple color when we are testing using Neidrin reagent. This we are going to do, and then we are going to see it in a while. And we also have what is called the biuret test. The biuret test is specific for proteins such as L-garbonin, casein, etc. In a while, we're going to see how that is done. Xanthoprotic test is a test that is specific for aromatic amino acids. An example of aromatic amino acids are phenylalanine, and your tyrosine, and your tryptophan. And also, we have the Million test. I'm sure you had that while you were in school, in secondary school. It is specific for phenolic group, such as tyrosine. We have Hopkins-Coles test also, which is specific for tryptophan, such as glycolic acids. And then we also have Sakaguchi test. Sakaguchi test. It is specific for amino acids such as arginine and then lysine, which have basic amino units in them. We also have what is called a lead acetate test, which is specific for sulfur-containing acids. And finally, we have the nitrocoside test, which is also specific for sulfur-containing amino acids, such as cysteine, methionine, and then cysteine. Let me also quickly remind you about the grouping of amino acids. Okay, let me also quickly say that amino acids can be essential or non-essentials. The essential ones are not synthesized in the body, and they should form part of our diet. the non-essential, maybe the synthesized in the body. The groupings of amino acids are into 7, but when we have the aliphatic, examples include glycine, alanine, valine, leucine, isoleucine. We have hydroxylic, an example is serine. We have sulfur, example is cysteine, and methionine. We have acidic ones, examples are aspartic acid, glutamic acid. We have basic, lysine, and arginine. We have aromatic and heterocyclic amino acids. Examples include phenylalanine, tyrosine, tryptophan, histidine. And then we have amino acids which are unique in their own way with their formation, their conformation, and their structure. And one great example of that is proline, and also its isomer hydroxyproline. Okay, now we have prepared some amino acids. These are amino acids that have been prepared that we are going to use for our tests, to carry out some of our tests. So please just stay tuned. We're going to go straight to testing for xanthropotic test. When we did our test on xanthropotic test, you remember we said it's specific for aromatic amino acids such as phenylalanine, tyrosine, and tryptophan. When we did our test, the test says we should put 2 mL of our solution, which is our amino acid. All the amino acids that are present, that we have shown you, amino acids that we have alanine, serine, glutamine, histidine, isoleucine, glycine, leucine, and all the other ones. And then the one protein that we have there is L-galbanine. We take 2 mL out of those amino acids, and then we add nitric acid to them, and then we heat in boiling water, and then we cool. When the solution is cooled, we add 0.880 normal ammonia, 0.08 ammonia, and then we shake, and then we look at the color. These are the colors that were obtained for the ones that are not aromatic and the ones that are not heterocyclic. Leucine is aliphatic. This is glycine, aliphatic too. Serine, hydroxylic, leucine, aliphatic. You can see that they are not positive for the reaction. They are not positive for the reaction. But we have the one that is specific. You can see this. This is tyrosine. You can see that the color has changed to something that's the color has changed. The color has changed. Tryptophan, the color has changed. This shows us that xanthropotic test is specific for aromatic and heterocyclic amino acids. Just stay tuned.